This recording is going to cover blood. Where would we be without blood? Blood is vital to our existence. It is the fluid that keeps us alive. As it travels through the miles of blood vessels, it transports oxygen from our lungs to the tissues of the bodies and removes carbon dioxide from the same tissues and delivers it to the lungs for elimination. Blood also transports hormones from our endocrine system, nutrients from our digestive system, and disease-fighting substances to tissues, as well as waste to the kidneys. Characteristics of blood. Color. The color is red and it approaches scarlet in the arteries and is sometimes darker in veins. Many folks seem to think the color of blood in our veins is blue. It's not, it's just the way it looks through our skin. The quantity. The average human body contains approximately 5 quarts of blood. Viscosity of blood is approximately 4.5 to 5.5 times the viscosity of water or thickness of water. Blood is relatively thick. Just keep in mind that blood is thicker than water. If you don't believe me, the next time you cut your finger, take a little blood and place it between your thumb and your index finger. Then bring both together and release. Now do the same with water. Do you see the difference? Blood is definitely thicker and more adhesive or sticky than water. The pH level of blood is 7.35 to 7.45, which makes it slightly alkaline or basic. This is important because most enzymes within our body function best at this range. You will also see this when you study microbiology and what effect pH has on the microorganism's ability to survive. The pH scale is something that measures whether something is acid or base or alkaline. The functions of blood. Blood has many functions. We discussed some of them in the opening paragraph, so let's summarize them to make it a little easier. It transports oxygen, carbon dioxide, food, waste, hormones. This is the responsibility of the red blood cells. It regulates body temperatures by removing heat from active areas such as our skeletal muscles and transports it to other areas of the body. Did you know that the temperature of blood is higher than our body temperature of 98.6 degrees Fahrenheit? It's 100.4 degrees Fahrenheit. Strange, isn't it? The next time you're in a hot environment, look at your skin. If it's red or flush, it's because the body temperature rises and it's diverted to the surface vessels. Blood contains buffers to control pH. These buffers help plasma resist any change in pH. It maintains water balance and a constant environment for our tissue cells. It defends against disease or infection, and this is accomplished by white blood cells. And it prevents hemorrhage, which is accomplished by our platelets or thrombocytes. The general composition of blood. There are 2 portions to blood or whole blood. 1, the formed elements or corpuscles, what we call our blood cells. This is the solid portion and makes up 45% of the volume of blood. And 2, plasma, the liquid portion of blood, which makes up 55% of the volume of blood. If formed elements are removed from blood, you are going to be left with plasma. Erythrocytes or red blood cells. These are the most numerous of all our formed elements. They are biconcave in shape, meaning they are caved in on both sides, thin at the center and thicker on the edges. This unique shape allows it to pass through the blood capillaries. Red blood cells only live for about 120 days because of the wear and tear of the plasma membrane, which is the outer lining of cell, as they squeeze through the capillaries. The function of an erythrocyte is to transport the oxygen or nutrients and carbon dioxide and waste. They lack a nucleus, which gives them more surface area to carry hemoglobin. The process of producing red blood cells is called erythropoiesis. The prefix erythro means red and poiesis means to make or form. This occurs in the red bone marrow of all bones until adolescence. Red bone marrow is then replaced with yellow bone marrow, which is fat bone marrow, in the long bones as we get older. After that happens, erythropoiesis occurs only in short and flat bones. In the fetus, this process also occurs in the spleen and the liver. Imagine approximately 3 million red blood cells enter your circulation every second. Red blood cells contain hemoglobin, which is responsible for the red coloring in blood. There are 2 portions or factors of hemoglobin. The heme portion is the non-protein portion and contains iron. This is the actual part of the hemoglobin molecule that is responsible for the red coloring in blood. The globin portion is the protein portion. Let's take a look at what happens when hemoglobin combines with oxygen, carbon dioxide, and carbon monoxide. Oxyhemoglobin is formed when oxygen combines with hemoglobin. This occurs within the capillary of our lungs. Carbaminohemoglobin is formed when carbon dioxide combines with hemoglobin. This is how it works. Oxyhemoglobin circulates to the capillaries of the tissues where oxygen is released and carbon dioxide is picked up by the hemoglobin molecule. Carbaminohemoglobin travels back to the lungs where carbon dioxide is released and oxygen is picked up. Carboxyhemoglobin forms when carbon monoxide combines with hemoglobin. An interesting fact that heme has a much greater affinity for carbon monoxide than it does for oxygen. When carboxyhemoglobin is formed, it deprives red blood cells of oxygen, causing headaches, dizziness, drowsiness, unconsciousness that can lead to death. You'll know if you have someone who has died of carbon monoxide poisoning, as a cherry red discoloration will appear in the tissues. Leukocytes, or white blood cells. These have a nucleus but lack hemoglobin, and they help our body fight infection. We can classify leukocytes in two groups depending on whether they have granules in their cytoplasm or they don't. Granulocytes, yep, you guessed it. This category of white blood cells has tiny granules in its cytoplasm. These granules are visible under a light microscope when stained. Take an egg white, think of it as the cytoplasm, and then take some sugar, your granules, and put them in the egg white. Now you can visualize what granulocytes look like. The white blood cells that fall into this category develop in red bone marrow called myeloid tissue. Again, your prefixes come in handy. Myelo means marrow. Granulocytes include the following white blood cells: neutrophils, polymorphonucleoleucocytes, or simply polys. These are the most numerous of all white blood cells. These digest harmful bacteria. Eosinophils, or acidophiles, increase in number in allergic conditions. They're going to release histamines. And basophiles increase during chronic infection and during healing from infection. The second group that white blood cells fall under is agranulocytes, meaning without granules. They do have some granules, but they are not visible under the light microscope, so therefore they are referred to as agranulocytes. These develop from both myeloid tissue and lymphoid tissue. Two white blood cells that fit into this category are lymphocytes. These increase if you have a viral infection, and these help form antibodies at the site of inflammation. Lymphocytes are going to be formed in your lymphoid tissue. Monocytes are going to develop from your myeloid tissue, and they digest cellular debris and foreign particles. Think of them as the cleanup crew. Now you may be wondering how your white blood cells help fight against infection and injury. Well, they are able to leave the bloodstream by way of a process known as diapedesis, which is the ability of a white blood cell to move through an unbroken capillary wall. Your neutrophils, eosinophils, basophils, and monocytes are phagocytes, which means they have the ability of engulfing and digesting foreign substances that are harmful to us. You're going to learn a little bit more about this in pathology during inflammation and repair. Platelets or thrombocytes. These are the smallest of the formed elements. They do not have a nucleus, and they develop in red bone marrow. They help in the clotting process and help form clots. Let's talk about an increase or decrease in blood cells. Erythrocytosis would be an increase in the number of red blood cells. Erythropenia or erythrocytopenia is a decrease in the number of red blood cells. Leukocytosis is an increase in the white blood cells, where leukopenia or leukocytopenia is a decrease in the number of white blood cells. Thrombocytosis is an increase in the number of thrombocytes, and thrombopenia or thrombocytopenia is a decrease in the number of thrombocytes. Did you notice that those suffixes osis, there's an increase, and those with penia, there's a decrease? The formation of various blood cells. Hematopoiesis or hemopoiesis is the formation or producing of blood in general. Erythropoiesis is the formation of red blood cells. Leukopoiesis is the formation of white blood cells. Thrombopoiesis is the formation of thrombocytes. Plasma. The second portion of blood is plasma. Remember, plasma is what is left after the foreign elements are removed from blood. It is a straw, amber, or yellow color. It is the stream in which blood cells travel. It carries not only blood cells but also nutrients such as sugar, amino acids, fats, salts, and minerals. It carries waste products, carbon dioxide, lactic acid, urea, etc. It carries antibodies, clotting proteins called clotting factors, hormones, and proteins. The components of bladder is about 92% water, 6 to 8% proteins, and they include your serum, your albumin, serum globulin, and fibrinogen. It also contains salts, lipids, and glucose. So what is serum? Serum is plasma minus the clotting protein. It is basically the fluid that remains after a blood clot has formed. Coagulation is the process of clotting or clot formation. There are substances in our blood that help stop the bleeding when we injure ourselves, as well as substances that inhibit clotting. If we didn't have substances that inhibit clotting, we would wind up with clots in the blood vessels and would eventually cut off our blood supply and oxygen to the various parts of the body. The substances in blood that promote clotting are thromboplastin, known as thrombokinase or cephalin, prothrombin, calcium, fibrinogen, and vitamin K. The substances in blood that inhibit clotting are antithrombin, antiprothrombin, or heparin. The process of blood clotting is very complex, but simply put, an injury to a blood vessel makes the tunica intima of the blood vessel rough. As thrombocytes flow through the roughened surface, they begin to fall apart and release thromboplastin. Thromboplastin can only clot if calcium and prothrombin are present. Prothrombin is made in the liver, but only if an adequate amount of vitamin K is present. Vitamin K is absorbed into the blood through our intestines. Thromboplastin and calcium act as an enzyme in a reaction that will now change the prothrombin into thrombin. This only happens when we have an injury. Thrombin is not present in the body, in the blood itself. Thrombin now reacts with fibrinogen to form fibrin. Fibrin is a thread of gel-like substances that form over the cut. The next time you cut yourself, look at the scab and you should see these little lines of threads. That's fibrin. Fibrin now entraps the blood, forming a clot. Have you ever noticed that there's a yellow fluid that oozes out of the cut? Well, that is what we call serum, the fluid that remains after a blood clot has formed. It is plasma, mining the clotting factors.